Assalamualaikum warahmatullahi wabarakatuh and good day to my fellow panel. Today, I will be presenting about my project proposal for PSM1 titled Gelatin Extraction from Fish Byproduct Using Different Temperature and Extraction Time for Biomedical Application. For introduction, my name is Muhammad Hilmi bin Zainudin. I am from program BMMA and I am currently working on this project under the supervision of T.S. Azman bin Ibrahim and my co-supervisor, Mr. Muhammad Saiful Firdaus bin Hussein. Without further ado, I will begin this proposal video with the introduction to my project. Firstly, my project research is about implementing gelatin from fish byproduct towards biomedical application industry. So, what is biomedical? Well, biomedical is a working in the biological industry which relates to the medical treatment. In the scope study of biomedical, there are a lot of medical treatment involved which uses biomaterials such as metals, ceramics, glass, and polymers. The use of biomaterials such as polymers are very common nowadays, especially in the tissue engineering application. This is because polymers contains wide range of physical properties and are adaptable to many applications such as drug delivery, medical devices, and bone tissue engineering. So what is tissue engineering? Tissue engineering is an application used in the medical treatment for organ replacement, bone grafting, bone regeneration, and many more. The application commonly uses polymers, that is craft with proper structure, chemical and mechanical needs towards the human body, which are called scaffold. This scaffold is then embedded in the human body as a substitute to provide cell growth and stimulate tissue regeneration. Now, to fabricate the scaffold, a biodegradable material is synthesized with the scaffold in order not to harm the human body when it is implemented. Natural polymers such as collagen, hydroxyapatite, and chitosan are biodegradable materials that can be used to fabricate this scaffold. Particularly, collagen contains gelatin, which share a very close molecular structure and function with collagen. Thus, gelatin is often used in cell and tissue culture to replace collagen for biomaterial purposes. Moving on, I will now explain to you about the problem statement for my project. Firstly, medical treatments specialized in bone grafting today are in a very high demand. It is also very costly, the procedure is very complicated, and the result is not very pleasing. This is because bone grafting is not the natural way of tissue regeneration, as it takes other parts of the bone from the human body to replace at the broken bone. And that there are limited source of bone supply to the industry, and this has become a global crisis with the high demand of bone treatments around the world, especially people in the golden ages. Moreover, the extracted gelatin commonly used in the tissue engineering application are mostly derived from mammalians. To exaggerate this problem even more, with the mad cow disease, nail and mouth disease, as well as religion boundary, has put a situation where we need a new source of gelatin that is capable to supply and meet the high demand of the people around the world. Now, I will explain to you about my project objective. The first objective is to analyze the potential of gelatin for fish bone and scale for bone tissue regeneration using bibliometric study. Next is to investigate the effect of temperature and extraction time towards gelatin properties. And lastly is to characterize the gelatin properties using viscosity, gel strength, and morphology. For my project research literature review, I will adopt the scientometric studies approach, which I will be collecting related published journals, books, academic papers, and other types of literature, with the aim of achieving a better understanding of the mechanism of this research. My main focus is to measure the impact of the related literature review as well as extracting important data to analyze the trend and understand the terminology behind my project research. By applying scientometric study, I will perform a bibliometric analysis using Scopus database, which I will prioritize keyword research, document citation, journal citation, and country citation. To perform the bibliometric analysis, I will be using Boss Viewer software, which include cluster analysis features to help me find pivotal articles and illustrate the linkage between related publications to my project. Other than that, I will also review the methodology of extracting fish byproduct gelatin from these pivotal articles to analyze the trend and summarize the result into a table which I will be used to formulate my own methodology for this project. As a result of my bibliometric analysis, you can find the data that I have tabulated in this slide. As you can see, there are about 5 total amount of publications that are related to my research studies. And as the trend, we can analyze from 2010 to 2020 the amount of publications towards my research project is increasing significantly. And the top 5 countries that produce the similar publications towards my studies are from China, United States, India, South Korea, and Iran. And this example of cluster analysis that I've done through Boss Viewer software. Now, for my methodology, I will follow this flowchart as a guidance in order to achieve my objective. The whole working process includes material selection, control parameters, pre-treatment procedure, type of solvent used, extraction temperature, and extraction time. Based on my bibliometric analysis result, I have chosen Pegasus catfish as my raw material which I will be used to perform my project methodology. The Pegasus catfish can be obtained from the Malaysian fish farming market as they are abundant source in this country. For the control parameters, based on my summary findings, the trend of using hydrogen chloride, sodium hydroxide, and citric acid shows a strong correlation between gel strength and the alpha chain content in gelatin. Therefore, I have chosen types of solvent as the control parameters for the gelatin extraction process. Besides that, referring to my project title, I will be studying the effect of extracting temperature and extraction time. Thus, the temperature and time are also the control parameters in this experiment. Now, I will explain to you the basic principle of extracting gelatin from fish byproducts that I will be performing for this project experiment. Firstly, the bone will be separated from the fish waste material such as the head, the fin, and the viscera. Then the bones will be scraped with a knife and washed with warm water to eliminate some of the flesh. After that, the sample will be minced using a piece of machinery. Then bone samples will be cleaned with tap water and drained off using cheesecloth. After that, we move on to the pre-treatment stage, which the minced bone will be soaked in hydrogen chloride to demineralize, followed by drain off and rinse with tap water. Then the bones will be neutralized with alkali treatment using sodium hydroxide. Then the step is repeated, but this time using citric acid as the alkali treatment. Next, we will move on to the next stage, which is the extraction process. The gelatin extraction from the bone will be carried out in distilled water at varying temperature of 45, 55, 65, and 75 degrees Celsius for varying times of 5, 7, and 9 hours. Then, extracted solution will be centrifuged at 10,000 Hg at 4 degrees Celsius using a piece of machinery. The upper phase is filtered with filter paper. The extracted gelatin will then be concentrated with rotary vacuum evaporator and dried in a freeze dryer. So that is how the basic principle of my project methodology. Now, for my result analysis, there are 4 characteristic properties of gelatin that I will be analyzing for this project research. The 4 characteristic properties are analyzed in order to meet my objective finding for this project, which are gelatin yield, gel strength, viscosity, and gelatin morphology. In general, gelatin yield increases significantly as the extraction temperature increases. The yield and functional properties of gelatin were dependent on the pre-treatment of raw material and the processing parameters, which include pH level, temperature, and time during gelatin extraction. Gelatin yield will be calculated using this formula: dried weight of fish bone gelatin over wet weight of ocean times 100%. Next, gel strength determination will be done based on British Standard 757/1975 method. The gel strength of the sample will be determined by using a texture analyzer. The results obtained are intended for comparison between packed catfish gelatin extracted at different temperatures. Moreover, to determine the viscoelastic behavior of gelatin gels, an oscillation rheometer is used. The viscosity is an important characteristic of the gelatin which will determine the grade and quality of a gelatin. The results obtained are also intended for comparison between Pegasus catfish gelatin extracted at different temperatures. Lastly, the surface morphology of gelatin will be analyzed by scanning electron microscopy (SEM) method. SEM uses an electron beam to scan gelatin samples to produce magnified image. The electrons in the beam interact with the gelatin sample, producing various signals that can be used to obtain information about the surface topology and composition. These signals are collected one by one or more detectors to form images which are then displayed on the computer screen. Based on the data finding that I will be collecting from my experiment, I have tabulated the table of results that I will be used to summarize my findings, like the table below. As you can see here, this is the table result for gelatin yield percent, and this one is the table result for physical properties of gelatin with different extraction temperature. And this is the table of results for physical properties gelatin with different extraction time. And lastly, are the SEM micrograph with different extraction temperature. That is all from me. Thank you for watching.